Let me kick off our first speaker, and it gives me great pleasure to introduce Professor Hedvig Hricak. Hedvig, to me, played an important role in my career. As an engineer moving over into the medicine field, when you switch fields, it's important in many ways to have an accepting community. And I never forget when I first met Hedvig and the role that she played in understanding the opportunities for engineering and the people associated with that. And I was completely new to this community. It was her and Sam that were very open and gracious. Hedvig was chairman of the Department of Radiology at Memorial Sloan Kettering since 1999 and is now transitioning her career. But she built one of the best cancer imaging departments in the world. Excellence in both the clinical research and clinical practice as well as research. And Memorial Sloan Kettering is a powerhouse, and they reached out to us at the University of North Carolina because they didn't have science and engineering, and we would go up there on a regular basis, and it was a great partnership over those years. She's considered one of the pioneers in establishing the clinical value of MRI for staging and understanding prostate cancer, kidney cancer, and gynecological cancers. She's an elected member of the National Academy of Medicine, and I can't imagine a better person I've been wanting to get her here since I got to here at Stanford. It's an honor to have you here. Dr. Hricak holds a Carol and Milton Petrie Chair of Radiology at Memorial Sloan Kettering in New York. Thank you for making the trip over. Please join me in welcoming Professor Hedvig Hricak.
Thank you, Joe, for this kind word. Yes, we had some interesting times together, and it was wonderful, every minute of it. It's a pleasure to be here, and it's a tremendous honor to actually speak in a symposium that honors Sam Gambhir. We all, I think everybody in this room knows, Sam was a very unusual person. It's not only vision, but his perseverance, his charm, and never give up in what you believe. Or like Winston Churchill said, never give up if you can think about something every single day. Well, that was Sam. Sam was advocate for integrated diagnostics. I know we had a symposium together in 2010 talking about integrated diagnostics. So let's speak about integrated diagnostics and the path towards precision oncology. I do need to show my disclosures. I serve on the board of directors IBA and Page AI, and both are very much pushing AI, machine learning. So it is kind of a part of the symposium, but there is no conflict. External advisory board for Johns Hopkins, international advisor at University of Vienna. I serve on both scientific committee and board of trustees for the German Cancer Center and scientific advisory board for Europe Bioimaging. So when we look at integrated diagnostics, although it has been around for more than 10 years, the idea that we need to integrate, there is still no consensus on the definition or scope. So to be flexible, there are different meaning to different people. Some, even today, will say that integrated diagnostics is just to integrate 2 or more diagnostic tests in the same specialty. Some say 2 or more diagnostic tests in different specialties, such as liquid biopsy and imaging, which is now becoming very popular. But ideally, and since a number of you are coming up from are here from the startup company, our long-term goal is to integrate all diagnostics and clinical domains of measurements. because unless you integrate them all, you don't know what you are missing. The integrated diagnostics finally had attention of the National Academy of Medicine because the number of tests that we have, not a single human being can really digest, and there are coming new tests are coming up weekly. So we had a workshop on incorporating integrating diagnostics into precision oncology care, and Gary Gold was a part of the workshop. There was a consensus, some. Diagnostics is moving toward data science, and everybody agreed that there is a need for data integration. There was a lot of talk of diagnostic silos that you spoke about. And one of the mandates was that there is an urgent need for collaborative initiative, and we simply have to eliminate those silos. Those silos are not only between different departments. There are a number of examples, silos within the same department between different sections. And then while building modern IT platform, the platforms that we have today, including glorifying Epic and everything, just will not make it for integrated diagnostics. But while building modern platforms, we need to remain flexible on the scope of integrated diagnostics and go forward one step at a time. Exponential increase in the number and complexity of diagnostic tests. We can no longer go like this, where we live not only in silos, but sometimes we shoot at each other saying which one is better. Together, we're going to be the best. And maybe ambitious, but I do hope that 5 years from now, we are all going to march in the same direction. We're going to be integrated, and the sea will be calm. But to do that, we need big data. We need algorithmic decision. And we really need a culture of collaboration. So integrated today, even two modalities in the same specialty, such as looking at MRI and the good old FDG PET. Patient has multiple myeloma, rule out bony metastasis. If each modality is read in isolation, then MRI is going to say they are very likely bony metastasis, low signal intensity in the spine. If nuclear medicine reads FDG PET, they're going to say there's no increase in uptake. There are no bony metastases. But if we work together, then we know that FDG PET is negative in about 11% of multiple myeloma patients. Those are bony metastases. We used to do biopsy. Now we do liquid biopsy just to see for the biomarkers and circulating DNA. And then that comes to the looking at CT, or cellular-free DNA in tumor follow-up. This is from our basket trial, because we know there is a pan-AKT inhibitors. Particular patient is breast cancer metastasis. There was serial C-free DNA in tumor follow-up, and then serial CTs. There is no question that the free DNA showed, the liquid biopsy showed decrease much sooner than CT. But that's not the problem. The question here is that such a correlation will allow us to update our clinical guidelines.
how often do you really need to do CT? We very often do CTs in clinical trials every month. Maybe we can go 2 months. So it's going to be more efficient, it will be better for patients, and it will be driven by new knowledge. A different example, circulating the DNA, seeing in a patient combined with the metabolic tumor volume and looking as a prognostic biomarker. Now, there's no question when there are circulating DNA as opposed to those patients that have negative DNA, that's going to be a big difference in the survival. There are no liquid tumors, no circulating cells, DNA. The patient is going to do very well. High number, they are not going to do well. But if we combine the metabolic tumor volume, then we can have a much more granularity. And for a patient, it means a lot. You have a negative, the DNA, circulating liquid biopsy, the DNA, but you have high metabolic tumor volume on FDG. You are not going to do that well. On the other hand, you have positive DNA, but you have very low metabolic tumor volume. You're going to do much better than if you look just at the positive DNA. And this is what that integration, even just between two diagnostic tests, gives you a much better prognostic biomarker. And then predictive biomarker. When we look at non-small cell lung cancer, the PD-1, PD-L1 is a miracle drug, truly miracle. But it is effective in only 15 to 20% of patients. Just think the other 80% that are hoping to be cured and nothing happens. But if you integrate simply radiomics, radiology to tumor volume, if you do integration together with the genomics as well as pathology, then you develop a predictive biomarker, and you can be much more sure that this patient is going to do well, so therefore is going to go on PD-L1. And this really, unfortunately, we need to look for other treatments. Predictive biomarkers in patient triage are so essential not only to save money, but look at the patient and what happens to a patient and the family. And then multi-platform omics and imaging. This is from Cold Spring Harbor. I love the study because 20 investigators worked for 1 year to have these results. There were regular liquid biopsies, but there was a regular manual tumor volume assessments, and then they only biopsied one lesion. And they said that integrating diagnostic data revealed new potential mechanism of response and resistance. The results were fantastic, but we cannot afford to have manual curation of the data, manual tumor volumes, plus we need a total tumor burden. And they said, unless we develop new analytical tools, we'll get nowhere, and we all agree to that. So the ultimate goal of integrated diagnostics is to look at all domains of measurements and data extraction. For the clinical notes, I think not only the natural language processing, but today LLM, the large language models, ChatGPT if you want, will give us an answer sooner than we think, but only if we collaborate outside medicine with our friends in computer engineering. Then we have radiology, pathology. Each of these measurements has a problem. Once we have data extraction, we need to do data aggregation. And then, most of all, we need to do integration and analysis and computer analytics. Only when we have the entire circle are we really going to have strong predictive and prognostic biomarkers and really reach precision medicine. So what are the bottlenecks? There are many. What are the challenges? The challenges that we have is, number one, let's start with radiology. My apology, but it's not moving. So in radiology, there are many challenges, but the three that I would like to highlight. For those that are not in radiology, it may seem strange that we are still asking for structural synoptic reporting, including standardized lexicon of diagnostic uncertainty. Then, of course, we need AI and machine learning for tumor segmentation, feature extraction, and classification. And we need automated anatomic and metabolic tumor volume and tumor burden. Many of you may say, we are there, we are not there. Structure of synoptic reporting. This is a transcribed report from May 1996. In many radiology practices, the only difference between 1896 and today is that report is no longer handwritten. When you look at structured and synoptic reporting in radiology, our publications are spectacular. We are building CV, but in practice, only about half of the practices in this country are using structured reporting, and synoptic reporting is even more rare. In radiology, we need more than synoptic or structured reporting. We need a lexicon, because what suspicious, probable, or possible means to each of us in the room is very different. So why not say metastasis present greater than 90%, 75, 50? We instituted lexicon in 2012. And I like to tell you an anecdote. At the faculty meeting, there was a revolt, but there was no option. And then one faculty say, I don't like it now. I have to think when I write the summary. Well, yeah, that's what radiology is all about. Then pathology. We always look at radiology being very technology-driven. But did you know that the College of American Pathology required synoptic reporting in 2013? Now, at that time, there were only 3 cancers: breast, melanoma, and colon. Their compliance was greater than 95%. Today, they added many more cancers to them. Genomics, a relatively new area, has structured reporting being distributed across our country, or this country, very much so, and being accepted and appreciated. So there is something wrong with radiology. We always look at surgery as not really being that technically acclaimed, but look at this. They are going to require synoptic operative reports by 2024, and they are going to require a 70% compliance. So other specialties are doing, and I think in radiology, somehow we have to wake up and really run with synoptic reporting. Oh, this is not an audience to talk about AI tumor segmentation. You may not agree, but I'll give you, as a clinical radiologist, also understanding what's going on around not only our country, but around the world. There's no question we need tumor segmentation. There's no question that there are many homemade, homegrown algorithms that really work. But in so many trials, in so many papers, and so often in a clinical work, we have a manual tumor segmentation. It is still used widely, and it is the greatest bottleneck to clinical and research advancement. And another one, radiology in AI and tumor segmentation and tumor characterization is more complicated than pathology. Pathology is running with AI. Quest is buying the prostate cancer. They bought algorithm. They're going to buy breast, and it's not inside information. It's all announced. But it is simpler because you ask algorithm for prostate cancer detection, and it's going to detect yes or no, and you're going to grade. In radiology, we need a multi-platform. We cannot just look at brain cancer. Or in the body, we need everything. We need lung, we need prostate. There's a prostate FDA, there are a number. There is FDA approved and FDA cleared. But that's not enough. We need for every site automated, algorithmic, validated tumor segmentation for every modality. And we need a multi-platform, intuitive reading and AI applications, not pushing button. Now I want stroke, now I want brain. No, we have to take a step further. We have to have a ChatGPT. Data governance. Joe alluded a little bit. In academic medicine, we do not have culture of collaboration, trust, and sharing. It has to be built. It has to be developed. With us, culture is strategy for breakfast, and sometimes even before. So what we need is sharing requires guardrails and protection for misuse data, but also misinterpretation. Protect interest patients. It's better we do it proactively than retroactively when there are troubles, faculty and institution. But we need mutual respect between specialties. So pathologists cannot read radiology and vice versa. I really think that data sharing requires guardrails and incentives. We don't share data, but then when we get money from NCI or foundation, and they require that we share data and they give us money as a award, then we all share. So there have to be incentives. Now, aggregation. There are many pretty aggregations being displayed, but integrated diagnostics extends beyond dashboards. Here I show you a very pretty dashboard on aggregated, user-friendly information display of prostate cancer. This is not integrated diagnostics. This is a dashboard, very clinically useful, because it really allows us to move through the clinics much faster. Dashboard data display is not integrated diagnostics. This is integrated diagnostics, multimodal computational integration, where we start with CT, the regular pathology, genomics, then we have a unimodal and then multimodal analysis. This is from our own work at MSKCC. So what we need in integrated diagnostics, we need technology stacks because there are different expertise that we're going to need to put in the development. And I love this quote from Shirley Tilghman: as much as new ideas are fundamental to the advancement of science, it is technological innovations that are the engine of scientific progress. And that's why we need that computational analysis, computational analytics, to truly have multimodal, integrated diagnostics of all domain measurements as predictive and prognostic modeling. And only then are we going to have truly precision oncology. Very briefly, clinical decision support is not integrated diagnostics. Integrated diagnostics is a part of the clinical decision support. And as we look at reducing medical errors, we need both integrated diagnostics, a part of that circle, but we need a clinical decision support as well. Definition of diagnostic errors is failure to establish accurate and timely diagnosis. That is integrated diagnostics. But equally important is communicate that explanation to patient or referring physician, and that clinical decision support is going to give us. So what did we learn? Multimodal data integration is a powerful approach to both clinical care and experimental data interpretation. I think we all agree. Multimodal data integration is a team effort requiring multidisciplinary and interdisciplinary collaboration. Retrospective data wrangling is a bottleneck to progress, and we really need to move forward with a prospective data. Investment in data engineering provides scale, automation, and standards, and it is absolutely essential for integrated diagnostics. Reproducibility in medicine, always hard, but we have to show it. And the big problem right now for many academic centers, I don't think it's the case at Stanford, it's really talented data scientists. Without talented data scientists, this will never happen. Opportunities, tremendous. I don't have to show you. Clinical AI-facilitated tumor boards. Very few places today have a luxury of multidisciplinary tumor boards taking time and being all together. They need to be AI facilitated. Research, endless opportunities for new discovery. Education with integrated diagnostics will be at different level. Patient access. We have to understand that today's patient is an informed patient. And the data shared and shared decision making is absolutely essential. You can no longer say, take 2 aspirins, call me in the morning. They'll ask you, why not 3 or 1? And health equity, it's a big item. But health equity will be enabled through integrated diagnostics and algorithms that you can use in the rural areas, you can use in the urban areas, and you can use it globally. Towards precision oncology, it's really reducing medical errors and improving outcome. So in summary, definition about integrated diagnostics, just one, it's not a CDS. Dashboards are not integrated diagnostics, and that's for the company representatives. Don't stop at dashboards, that's not enough. And long-term multifaceted benefits of integrated diagnostic, that's for the hospital administrators. It's very difficult to justify. It's not a P&L. It's very similar what we had in PACS at the beginning, and it's a new model. Why do we need better outcomes? Steps, interoperability. I know Mike is here, but we still don't have, of systems and standards. Synoptic reporting, we really need to move there. Annotation, segmentation. We have to move and give ourselves deadline from manual to augmented to really unsupervised and automated process. It is difficult for clinicians to trust automated process, but we don't have a choice. Culture, we talk plenty about that. And what I really like, we need to really put efforts in training a new generation of integrated physicians and scientists. So is integrated diagnostics today a myth or reality? Well, I think that we are able to look at the egg, paint the bird, because we do have vision, interdisciplinary collaboration, and perseverance. And with that in mind, thank you very much for listening.
What I found wonderful about your presentation was the clinical grounding of this call for integrated diagnostics. I mean, there's a richness in understanding the value of what that's needed for those of us on the research side that I think was very compelling. Let's open it up to questions for Hedvig. There's a microphone up here.
Hi, thank you so much. Great talk, I appreciate it. I'm not a clinician or a PhD, I'm still a regular kind of guy, but how do you envision like liquid biopsy and molecular imaging playing together, kind of practically in the field? Because it could be, you have liquid biopsy positives for 5, 10, 15 years, but then there's no solid tumors. Like how would you practically handle that?
Are you asking how do we envision the relationship between liquid biopsy and the biopsy of solid tumor biopsy, the regular, right?
Correct, yeah.
Okay, so there's no competition. Every time you do a tissue biopsy, always remember there can be problems. Patients don't like to have, wouldn't like, they sign for the tissue biopsy every 3 weeks, but they're not going to sign up for every week. Liquid biopsy, the blood test, you can take every week. So number one, the frequency of the two is very different. Number two, while we are very early, now we are talking about metastatic disease. Metastatic disease, the value of liquid biopsy is different than in the detection, new detection. That's what Sam had an idea for ovarian cancer. I'll take blood and I'll detect. So let's talk about metastatic disease. In metastatic disease, we already know for number of cancer, there is no recurrence unless liquid biopsy is positive. Myeloma, there's no recurrence unless you have a liquid biopsy positive. So the liquid biopsy is extremely powerful. I think we're going to, as we already see, start with liquid biopsy follow-ups. When the liquid biopsy is positive, then we're going to move to imaging to see where the disease is. And at that point, we need targeted imaging. CT is wonderful, but moving forward, we need more than CT. We need targeted imaging to the biomarkers we are looking for. Not FDG PET, don't get me wrong. It's wonderful, it has revolutionized our oncology care, but it is not tomorrow. Tomorrow, we need PSMA for prostate cancer. So we need much more targeted molecular imaging. And that's why in radiology, the big next is molecular imaging and targeting diagnostics. So that's where we go. But there is no, it's actually going to be sequential. We're going to have clinical guidelines to know what to do when.
So like in the case of like a GRAIL, it's like say a consumer, like a Palo Alto person.
Oh, I know.
Like they're taking a GRAIL test and it's, you know, the percentages aren't 100% on that. And then like, how would you track, like if we're thinking about like a consumer directed patient care, like, so obviously the radiology center doing MRI screens is the one with a lot of press these days, but I think there's a lot of, maybe not, it's not a great test possibly for all cancers, of course. So I'm thinking like a good consumer, quote unquote, consumer strategy would be like GRAIL, like, or there's a company in Southern California I'm familiar with called Hawkeye. They do like a blood test, and then you kind of can track that over time, and then yeah, as maybe the percentages increase or statistics increase, then you can actually trigger in a PET scan, and then you could actually trigger in like a chemo or smart diagnostics agent on that. Yeah.
So I think that we have to be careful with GRAIL as well. There is a difference in the value at present of liquid biopsy for screening, early detection, and detection of the recurrence. When we talk about detection of recurrence or metastatic disease, you also have to know for which cancers does it work. So it has to be, it's a long process, but it will happen. We don't know if it really works for all cancers. And that's what we need to, we just need to be sure before we advertise. Because you have a patient's hope behind it. And it can be like for screening breast cancer, you know there was a problem. And the hope that it was very high and then it went very low down and it's going to level out. But that's why you're dealing with people. And that's what you always have to keep in mind before you release any press release or before you make a claim that may not really work. But don't worry, it's not only liquid PD-L1. We thought we're going to cure lung cancer overnight. Well, not so fast. Immune therapy, CAR T cells, oh, magic, not so fast.
Thank you, Connie Chu from Orthopedic Surgery. Wonderful talk on integrated diagnostics, which I think have broad appeal. And I was wondering if you could tell us what your first effective step was in building your integrative diagnostic team.
So the first one, and I can tell you it's not easy, get everybody in the room and agree to share the data. That's number one. We use the uniform fabric. So you have to have a technology that allows that data sharing and aggregating it on the same platform. But then the second one for each specialty is little different. I showed you problems with radiology, but there are problems with pathology. Just digitizing the imaging is not enough. And both need both reports and image digitizer in order to aggregate and then to be able later on to do analytics. Everything has to be quantifiable. You cannot aggregate little better, slight progress. So there are a lot, and you have to work very closely with your colleagues in computer science and engineering. But we do, and what we do, one cancer at a time. So we are doing brain cancers, ovarian cancer. You cannot do all cancers at the same time.
On that thought, you spent time at a medical center that was broadly defined, and now you're at a cancer center. Do you find the level of integration and the approach to integration fundamentally different with the focus that you have at Memorial Sloan Kettering that you've seen at other academic centers?
Yes. Yes. I worked at UCSF for 18 years. I always, no question asked, I only wanted to do oncology. So when I was sent to San Francisco General to do trauma, I was miserable. Because one fracture, two fractures, three. So anyway, no, I just joke. But I always had passion. And in an academic center, the competition between infection disease, cardiology, neurology, and cancer, there is a competition. And there are only that many people, in our case, radiologists, that can go around. So it is much more complex. Now, we should ask Gary, but most of the department now do have oncologic section, imaging. At Memorial, because we always say we all breathe, dream, and work oncology. So we have very much more unity and the common sense of collaboration, because we are not looking at Alzheimer. We only look at cancer, so it is easier.
My name is ZJ Yang from radiology. I'm a big fan of the integrated diagnostic. Actually, I have done research combining liquid biopsy with imaging for cancer diagnosis. I'm actually curious about your thoughts because recently the multi-omics really trying to combine biomarkers or important features from different categories for diagnosis. But I guess for some tools, let's say for sequencing, we generate so many of the biomarkers, like fully understanding their role in pathology in each different cancer can easily take a lot of time, right? But if we don't fully understand them, do you feel safe to use them directly in clinical? Or like to what extent that we have to understand its role before we really put it into the clinical scenario? Kind of curious about that. Thank you.
You're right. That's what we talked a minute before that, and that is putting things in the clinical practice before you fully understand. And as you know, the 10 years that we say for 2000 to about 2010 was the decade of genomics. And we thought genomics are going to give us all the answers. Then Henry Weinberg came out and said, oh, excuse me, guys, it's all about metabolism. So then we thought metabolism is going to do. So you're going to see those steps. Now we are in the decade of metabolism. I'm only talking about cancer. That's all I know. And that's why we have the journal, Metabolomics, and all of this. So I think it's just the time will tell. It will self-correct. You know that genomics are wonderful. And omics are wonderful, but there are very few targeted treatments that work strictly on genomics. And there are many more excellent drugs coming out that are targeted beyond genomics. And that's why you need integrated diagnostics. I have to say thank you for the question. Sorry, I didn't emphasize. The key is well-curated data. Absolute key. You have bad data in, bad results out. So that data curation up front, let's take radiology, but it's the same for pathology. Those data have to be well curated, not only in volume and radiomics, but interpretation as well. And this is why it has to be a team. If you don't have radiology engaged in quality control for data curation before it goes into the second part of the aggregation, nothing will happen. So that's where I said you cannot read pathology and pathologists cannot read radiology. It's quality of interpretation and data curation. Well, we have a number of examples where the large data sets fail because they were not well curated. And we know that Flatiron, that really did very well, spend by far the most money on data curation. So those are the real world examples. Data curations and Flatiron I don't know if anybody's here from, but we really look up to Flatiron, how much they put in quality, data curation, and quality of data.
Daniel Kapp, radiation oncologist. Thank you very much for an excellent talk. Can you comment your thoughts on the biases in your data sets? You're working at an institution that's fairly selective in your patient population, and when you work with IBM, it produced really results that were not reproducible in other real-world situations. Can you comment on how you're dealing with this type of bias?
It was MD Anderson, not a Memorial Sloan Kettering. So just the data that were questionable afterwards. Yeah, that's what we believe. But I'm not so sure I should talk much about IBM and why Watson failed. But I can tell you, I hope nobody's here, and this is not recorded, they didn't listen. They knew it all coming in. Sorry. And we actually, in radiology, I withdrew from collaboration way, way before. Nobody could believe that we withdrew. I withdrew from radiology from the IBM, because they didn't listen.
My concern is not just on that one specific case.
Yeah.
It is that most databases in radiology come from several select institutions. And I'm wondering about their applicability in a more general situation.
Okay, you are right. But cancer is cancer. Stage 4 lung cancer is a stage 4 lung cancer. Our advantage is that we have such large data sets in every stage of the cancer. But stage 1 lung cancer is the same at Stanford, UCSF, Iowa. So you have our advantage, and that's why PAGE AI, prostate cancer, works, because we had a huge data set. The larger the data set, well-curated data, the better the results.
Thank you.
Okay, on that note, please join me in thanking Hedvig for a very inspirational presentation. Thank you.